So I'm going to be talking about human immunity, covering non-specific defences against disease, specific immunity and the biology of vaccination. So, non-specific defences against disease consist of the first and second lines of defence. So the non-specific defence systems is known as the innate immune system, so it doesn't distinguish between pathogens and responds to them all in the same way. So pathogens are infectious agents that cause an immune response in the body. So they can be cellular, living, or they can be acellular, non-living. See figure 1. So the first line of defence is physical and chemical barriers which prevent the entry of pathogens into the body. So they defend the body from infection. The skin is the most important physical barrier and it covers almost all parts of the body. And mucous membranes are a mechanical barrier to the wet internal part of the body exposed to air. So they line the respiratory, digestive, urinary and reproductive tracts. So the skin is comprised of 3 layers. The epidermis, the outermost layer, it's impenetrable and it's composed of tightly packed cells preventing entry of pathogens. And it also creates melanin, so it protects the skin from the sun's UV rays. The dermis is underneath the epidermis and the subcutaneous is the innermost, deepest layer. And hairs on the skin block dust and dirt from settling on the skin. Sebum is created by glands in the skin and it gives it a slightly acidic pH, so it forms a waterproof chemical barrier and prevents microbes from growing. Mucous membranes consist of a layer of epithelial cells which secrete mucus, so it's a sticky fluid which traps pathogens and performs mechanical actions to flush microorganisms out of the body and away from potential sites of infection. So the nose, mouth and eyes are entry points for pathogens. So tears, mucus and saliva all contain enzymes that break down bacteria. Any pathogens that are not killed get trapped in the mucus and then swallowed down into the stomach. So the nasal cavity and airways contain cilia, which are hairs which trap invading microorganisms and dust so that move particles away from the lungs. Urine flushes out bacteria from the bladder and stomach acid kills bacteria and parasites that are ingested. Now the second line of defence is known as the immune response. So if pathogens get into the internal environment, tissues, cells and organs in the immune system work together to protect the body and attack these pathogens. This is known as the immune response. So antigens are the part of the pathogen that tell the body it's being attacked. So they are what causes the immune response. Antigens can be protein, carbs, nucleic acid or fat. So the body combats antigens to stop an infection. The inflammatory response is the body's first defence in response to cellular damage. So, inflammation fights infection by destroying the pathogens, confining the infection to one location in the body and repairing damaged tissue until the infection or injury is healed. So, inflammation can be acute or chronic, and it shows as redness and heat due to an increase in blood circulation, swelling, which is an increase in blood and proteins to the area, and pain due to mechanical pressure on nerve cells in the damaged area. So the inflammatory response, figure 5, is triggered by mast cells, so they release cytokines and histamines when tissue is infected or injured. So these chemicals communicate with each other and with other cells to coordinate the inflammatory response. So macrophages detect, destroy and engulf damaged, defective or invasive cells. So when inflammation is present, the release of chemicals, histamine, from the damaged cells causes monocytes, which is a type of white blood cell, to differentiate. So they leave the bloodstream, concentrating at the site of infection and become specialised cell-eating macrophages or phagocytes. So phagocytosis in figure 6 is a process in which the macrophage engulfs invasive cells in a phagosome, so it's a vesicle. Once engulfed, enzymes inside the macrophages destroy the ingested particle. So the phagosome then joins with a lysosome, which is a vesicle containing digestive enzymes, forming a phagolysosome, which digests the contents of the phagosome. So macrophages can modify themselves to form different structures to combat infections. So fever. At the area of infection, there's an increase in body heat. So this helps to combat the growth of pathogens by the inactivation of enzyme activity. And it can also increase the metabolic activity of body cells to strengthen the immune response. So damaged cells and platelets release chemical signals called clotting factors. So they allow platelets to become sticky, forming a solid plug known as a clot. So this prevents entry of foreign pathogens and also prevents further blood loss. So specific immunity. The third line of defence, also known as the acquired specific immunity. So it involves cells and chemicals which recognise the pathogen invading specifically, targeting specific antigens and acting against them, formulating an immune attack. So acquired immunity isn't present at birth and it develops over a lifetime. So it's learned from the person's immune system experiencing the disease, with responses, antibodies quickly being made and with greater force once it's recognised. So it can be active or passive, and we'll discuss this further in section 3. Now, the roles of the different types of immune cells. So white blood cells, also known as leukocytes, they're made in the bone marrow and are part of the lymphatic system. So they seek out disease-causing organisms or substances and launch an immune attack. So there's 3 types: granulocytes, lymphocytes and monocytes. So granulocytes contain granules, contain small proteins, which are basophils. So they contain histamine, which dilate the blood vessels during an infection, so they're present after an allergic reaction. Eosinophils are rare, so they play a role in the general immune and inflammatory response in the body. So they release toxins from their granules to kill those pathogens. They're common in allergic reactions and parasite infections. Neutrophils are the most abundant and are known as phagocytes, scavengers, which surround and destroy pathogens. So they multiply early during an infection and they're the first to arrive at the site of infection during inflammation. So lymphocytes are made in the bone marrow. Once mature, they create a defence system catered to specific pathogens in the acquired immune system. So we have B cells. So they mature in the bone marrow and are part of the humoral response of the acquired immune system, which means they produce antibodies to help the immune system mount a response to infections. So they become activated when they detect foreign antigens with their receptors or receive signals from the helper T cells, which we'll cover. They clone into mostly plasma cells and memory cells. So plasma cells produce antibodies, and as long as there are plasma cells, immunity is maintained. So B cells don't leave the lymph nodes, only the protein, the antibody bits, move around the body via the bloodstream. So T cells, they make up 80% of lymphocytes and they're involved in the cell-mediated response of the acquired immune system. They recognise and remove infection-causing cells and leave the bone marrow soon after they are created and mature in the thymus. So they differentiate into 3 different types of T cells. So they each contain different receptor molecules on their surface and they're released into the bloodstream at the site of an infection. So cytotoxic T cells combine with antigens. They find and destroy bacteria, viruses and cancer cells by creating a protein known as perforin. So they perforate the infected cells, releasing enzymes which causes self-destruction. Suppressor T cells suppress the cytotoxic and helper T cells to stop them causing excess damage to the body's own tissues once antigens have been dealt with. So it's important in protecting against an autoimmune attack. Helper T cells, the most common type, regulate all immune functions within the body. So they release lymphokines that stimulate other types of T cells to grow and attack, and they also activate B cells, helping them grow and mature. Now monocytes, we've covered in section 1. So they make up around 2 to 8% of white blood cells. So they are the largest of the white blood cell and they target and destroy infections. So protein molecular defences are proteins produced by other proteins in response to the presence of foreign material. So they work with antibodies to trigger a complement reaction, shown in figure 8, that either attacks invading microbes directly or hinders their ability to reproduce. So they protect the body by, so they destroy pathogen cell membranes, they recruit phagocytes to the site of infection, they identify pathogens, and they intensify the immune response by inflammation. So this causes further immune cells to the site. So cytokines are small proteins, their presence signals more cytokines to be released. Now antibodies are defensive proteins that bind to antigens. So their biological functions generate protection and eliminate pathogens which are harmful to the host. So they prevent pathogens crossing the placental barrier, they activate complement proteins, they neutralise antigens, they join phagocytic cells, and they act as osponin. So they are structurally composed of 4 polypeptide chains, so 2 light and 2 heavy, joined by disulfide bridges. So there are 5 types of antibody as seen here. IgA provides a primary defence of inhaled and ingested pathogens. IgG is the most abundant, and it's a major activator in the complement pathway, attacking anything it deems as foreign, the only type of antibody transferred passively through the placenta from mother to fetus. You've got IgM, the largest antibody. It's got a different structure, and it's the first to be synthesised in response to pathogens. IgE is the least prevalent and increases significantly in allergic reactions, causing recruitment of eosinophil at the site of infection. And you've got IgD, a B cell receptor thought to be involved in the humoral response. So it regulates B cell selection and homeostasis. Now, hypersensitivity is what is known as sensitivity to antigens. So the immune system doesn't display the same level of sensitivity to all antigens. So when defence mechanisms fail, excessive and undesirable reaction is triggered in response to repeated exposure to the same antigen. So autoimmune disorders are one of the causes of hypersensitivity. So immune responses are directed towards self-antigens. So these reactions can't be escaped because of the positive feedback loop. So hypersensitivity diseases tend to be chronic, unfortunately. So the types of hypersensitivity we've got immediate hypersensitivity type 1 involved in anaphylaxis and allergies. Antibody-mediated type 2 causes inflammation, phagocytosis or functional derangements. You've got number 3 is immune complex reactions, which are common in systematic lupus and chronic autoimmune disorders. And number 4, cell-mediated immunity is mediated by helper or cytotoxic T cells, and diseases include Crohn's and MS. Now, the MHC complex is the most important system the body uses to distinguish between self and non-self. So there are 2 groups of MHC markers on the plasma membranes of almost all body cells, and it's a collection of glycoproteins all unique to the individual. So MHC1 is when a cell becomes infected, unfamiliar proteins are synthesized inside the cell. So small segments of the antigens are expressed on the surface of the cell, which indicate a non-self cell apart from red blood cells. So MHC2 markers are present on antigens from extracellular pathogens. So ACPs break down the antigens and incorporate pieces of them with the MHC2, which is recognized as a non-self. Now, self-tolerance of own tissues is antigen-specific immunological unresponsiveness. So a normal immune response consists of recognizing a harmful antigen, activating and mobilizing forces to defend it, attacking it, and then controlling and ending that attack. So if the immune system malfunctions and mistakes self for non-self, it can attack its own tissues, which causes an immune disorder, so the failure of self-tolerance. So no one knows why self-tolerance fails, but it is suspected that a combination of tissue damage or susceptible genes may be the answer. Now, rejection of transplanted organs occurs in all transplant recipients to varying degrees. So the immune system recognises the donor organ as foreign, so it triggers an immune response. So hyperacute rejection occurs minutes after the transplant, so when antigens are completely unmatched. So if the organ isn't removed, the patient will die. Acute rejection occurs anytime from the first week to the first 3 months, and chronic rejection is a longer time, slowly damaging the donor organ. So if antigens are very similar, it elicits a much weaker immune response. So this is why siblings or parents are often the best donors due to the genetic match between the MHC genes. To decrease the risk of rejection before transplantation, we try to ensure that recipient and donor have compatible blood types, so relatives are preferable. After the transplant, patients should take anti-rejection medication consistently, and lab results should be monitored frequently to check for changes. So autoimmune disorders are very common, so they affect a single organ or cell type. At present, there isn't a cure, but remission can be achieved in some disorders. So management of symptoms decreases the severity. The following are practical actions that can be taken to help alleviate symptoms. So eating a balanced diet, exercise, rest, taking supplements, decreasing stress, and limited time in sunshine, and just avoiding any known triggers. So rheumatoid arthritis is a chronic inflammatory multisystem disease in all joints characterised by destructive synovitis, mainly in the small joints of the hands and the feet, see figure 11. Now, joint inflammation and pain shows in the blood concentration as elevated levels of C-reactive protein in synovial fluid and the formation of autoantibodies that attack the joints of the organism. MS, multiple sclerosis, affects the nervous system in which immune cells intermittently attack proteins in the brain. So it causes repeated episodes of inflammation within the brain. So it affects sight, mobility or sensation, followed by periods of recovery. So the body can't distinguish healthy tissue from infected tissue. Genetic, environmental and stochastic elements contribute towards developing MS. And as time progresses, attacks can lead to permanent disability or scars. So inadequate regulatory function is associated with loss of inflammation, insulation on nerves and secondary loss of nerve cells, sclerosis. So see figure 12 for that. Now, section 3, the biology of vaccination. So there are two types of acquired immunity, active and passive. And these can be either natural or artificial. So active is the most common type, and this is long-lasting immunity. It's developed after being exposed to a specific type of pathogen. When the same pathogen is encountered again, the memory cells recognise it and immediately activate the rest of the immune system to fight the pathogen to prevent infection. Passive immunity develops from the receipt of antibodies from somewhere or someone else, and it's short-lived, so it doesn't cause the immune system to recognise it again. There's no memory cells. So artificially acquired immunity involves human intervention, for example, a vaccination or antibody treatment given for a specific purpose. Natural acquired immunity is exposing the body to an infection or maternal antibodies. So natural immunity is essential for protection against a variety of infections the body is exposed to. Now, vaccinations. So they contain weakened or inactive pathogens that act as antigens. So they trigger an immune response within the body to produce antibodies towards the pathogen. If the body encounters a pathogen that has previously been encountered, memory cells recognise that antigen. This is called immunological memory. So the immune response occurs more quickly and more effectively than the first encounter, which is why vaccines are used to protect against targeted disease, see Fig 14. So live vaccines contain viruses or bacteria that are specifically treated and weakened to make them harmless. So they mirror the body's natural response to infection, creating a broader immune response, so normally only one dose is needed. And inactivated vaccines are made by growing bacteria in a culture media and activating it with heat or chemicals. So this requires multiple doses to allow the body to produce long-living antibodies and develop memory cells. Now, the benefits of vaccination for measles. So the first vaccination was introduced in the UK in 1968, with the MMR introduced in 1988. So since it was introduced, infection notifications in the UK, see figure 15, have drastically declined, which shows infection rates between 1940 and 2018 when it was eradicated. Now, the benefits of the vaccines for measles include thousands of hospital admissions have been avoided, thousands of deaths have been averted, it protects others that might be unable to have a vaccine, it can save family time and money from long-term care associated with prolonged disease, and it protects future generations as passive immunity occurs during pregnancy. Now, vaccinations protect individuals from diseases, but not everyone can be vaccinated. So people with weak immune systems or people with allergies. So the more people who are vaccinated, the less likely people who are unable to be vaccinated are of being exposed to those pathogens. This is called herd immunity, see figure 16. It does not provide full protection, but everyone has substantial protection thanks to vaccines. Now, vaccines have been successfully developed to provide the best defence for a number of diseases. So in the UK, they are made to be convenient and accessible. And individuals that are at risk are also provided with accessible vaccines. So the factors affecting uptake are not giving a vaccine to someone who is recovering from another illness, anti-vaxxers who don't believe in them, cost of vaccines, pregnant women can't be vaccinated, and perceived efficacy of a vaccine. So older people are more likely to be vaccinated, and some people are more willing to affect. So, the summary, non-specific defences include the first and second line of the defence, the innate immune system. It doesn't distinguish between pathogens. The specific immune system, the third line of defence, involves cells and chemicals which recognise the pathogen invading specifically, targeting specific antigens and acting against them. Vaccination triggers an immune response within the body to produce antibodies towards a pathogen, so they're given for a specific purpose, rapidly fighting infectious agents before symptoms manifest, so they're hugely beneficial. Thanks for listening.